All right, so main ingredients of beer: grain, hops, water, yeast. We're starting with grain. All right, we're going to start with grain. Grain itself that we use is barley. That is the most common type of barley. Oh no. Take that shot. Let's see if this one will work. All right, so a barley grain. There is composed of a couple of different parts. All right, it's a seed. That's what it is. Make sure I'm up here. Barely see it. That's okay. We're going to talk about the key parts of our barley grain. All right, so on the outside is the hull. And so this outer part is the hull. We do use a part of that. All right, when we brew, the hull itself provides a nice filter in our mash. Wheat doesn't have a hull. The wheat grain doesn't have a hull. So if we brew with wheat, we have to consider the possibility that we have to add some extra stuff just to help us filter all of the grains out of our mash tun. Inside of that hull, we have the actual seed itself. All right? So this is, you have our hull, and then the actual seed, or the embryo. That's what we can talk about. This embryo has, or the seed itself has, the embryo proper. This is the germ. And it's called the germ, which is basically the embryo. And then it has the rest of this that's called endosperm. This is going to be providing the nutrients to our developing embryo. On the surface, just under the surface, is a layer called the aileron layer. Aileron layer. All right, so what a seed is going to do when it germinates, all right, we're going to add water to it, it's going to germinate, that's going to stimulate a series of processes where, starting at the germ, we start having what's called the acrospire growth and the rootlet growth. So your acrospire is your shoot. And you may have seen some of this in another class, like in a biology class in high school or maybe middle school, where you may have soaked beans or radishes or something like that in water, and then you come in the next day and you see one, maybe both of these things sticking out of a seed. That's all coming from the germ. And it's a plant, so the plant's going to have to photosynthesize, but during the first parts of its life, it can't photosynthesize. It doesn't have the chlorophyll in it. It doesn't have, in many cases, it might be below ground. It's not seeing light yet. So this growth is going to utilize the stored energy inside that seed. And that seed has this endosperm. The endosperm itself is made of starches. Made up of starches. All right? But the starches can't really be used yet. If they could, nothing would prevent these seeds from just randomly sprouting. We know that doesn't happen. You buy a pack of seeds, or you have grass seeds, or vegetable seeds, they sit in a packet and they just stay there, right? They need water. Why do they need water? They need the water because we have to activate a number of enzymes to start freeing the sugars from these starches, and it's those sugars that are then broken down and utilized for energy. So, what all goes into this? We have oils and lipids in this aileron layer. It has minerals. It has minerals that the developing plant can utilize. It has plant or seed. Has about 30% of the seed's proteins in this layer. Right, so you've got these compounds, oils, lipids, those can be serve as energy molecules too. Fats and lipids, very high energy. Proteins could be very high energy. And then you have these minerals that are kind of stored in there. So if it happens to land in an area where those minerals aren't immediately available, they have them to start growing. But this layer makes something called alpha amylase. Alpha amylase. So in the process of germination, water will start to soak into the starches, start to soak in the seed. That water will start to activate another enzyme called the beta amylase. Once that beta amylase starts to become activated, it then triggers the production of alpha amylase, all in this seed. Those two amylases will break down the starches and convert them into sugars. Now, how did these things get activated? All of this inactive beta amylase. Well, it gets activated by the germ itself. It releases beta amylase. It releases some beta amylase, starts the process, and then it starts secreting a chemical to say, hey, we need this to be activated. This guy is important to have because that's our signal. So we're using just a natural process of the seed to get these enzymes that we need, the alpha and the beta enzymes. And that is the whole reason why we have to malt our grain. We have to put it through a malting process. The malting process is simply activation of these enzymes. Now, here's the thing. If we let this grow for too long, it's going to consume all of our starches. All right, is that a bad thing? Yeah, it is. Because what's our sugar source? It's from our starches. So during the malting process, what we're going to do is we're going to germinate these grains. Then once they germinate, we're going to stop it. We're going to stop it by heating and drying it out. The alpha amylase is still present. It's active. The beta amylase is present, active. We have a bunch of starches. Why aren't these things still working on the endosperm? Why aren't they still breaking it down? It needs water. It needs to be in water. All right? So that's our seed. Let's talk about these amylases and starches. All right, starches exist as a chain. All right, a starch exists as a chain, and there's a couple different types of chains and branches and so forth, and they have some various names. All right, so if we look at a starch, we've got something that kind of is going to be branched, very big branches usually. Some of them will have branch points all along. Some of them, our branches, might not have a branch point. So a starch could look something like this. And a starch that looks something like this is, I believe it's called amylopectin, would be a term for it. All right? That's how it will work. If we were to take a look at something like this, blow this up, to look at it a little bit more closely. Our starches are basically a series of glucose molecules. Let's get this over here. So, it's a series of glucose molecules that are all linked together. They're all linked together. Glucose is basically sugar. It's the main energy source of cells. All right. Tidbit, our brain uses glucose. So when you have low blood sugars, you might get into a mental fog until you adjust properly. If you try the keto diet, you're going to be on it for about 2 weeks until your body adjusts and adapts and starts creating its own glucose, its own glycogen, I should say. But yeah, glucose, series of glucose. Right now, we can't use it. The yeast can't use it. It's in this form that's just inaccessible. This is where our amylases will come into play. Our amylases will break these glucose molecules, it will break the starch molecule into usable glucose chains. All right, so how does it work? Alpha amylase. Let's do this. Alpha amylase. Let's do this. Random cleavage. All right. So alpha amylase is going to exhibit random cleavage. What that means is alpha amylase is going to come in here and it's just going to go, I'm going to break there, I'm going to break here, I'm going to break here, I'm going to break here. This is an alpha breakage, all right? And what we're doing is slowly breaking it down. Sometimes the breakage will result in a 2-glucose sugar molecule. This is maltose. That's usable by our yeast. Sometimes it cuts it and it produces a 3-chain. This sugar molecule is called maltotriose. Maltotriose. Not all yeast can use it. I'll say most of them can, but not all yeast. Sometimes it'll break into a single molecule, which is glucose, which is super easy to use. Other cases, it'll break it into some accessory sugars, other types of sugars. But alpha amylase gives us this random cuts. So we can still have big, long chains of starches, or we can luck out and get some of these. Beta amylase is what I'll refer to as Pac-Man. Beta amylase will start at the end of these chains and chop them up. And there's some requirements, some restrictions on it. All right? So Pac-Man's gonna come in, it's gonna take a chomp, but it's only gonna chomp out 2 of these molecules. So we get 2. And then it continues, the next 2, and the next 2, and the next 2, until we get to a break point, and then we stop. We can't do anymore. And so beta is going to keep doing that. Anywhere where alpha cuts opens up a new chomp point for our beta amylase, for our Pac-Man to come through. All right? So what we've done, just kind of as a summary, is we've taken this grain, wherever all the enzymes are inactive, we have a lot of starches here, we germinate it, so we activate our amylases, make them so that they can work, and then we dry it out and we stop that malt, so that now when we add sugar, when we make the mash, we add water, hot water, and get everything dissolved, now we're letting the alpha and beta amylases take these long complex chain starches and start breaking them up into glucose, maltose, maltotriose. Now there's going to be some things that are left. All right, you're going to have some branches, some branch points that are there. Other enzymes in our mash, other enzymes found in the seed are going to be important in breaking some of those down. All right? Questions? Now, we use this in the mash. Any brewer uses this in the mash. Oops. Let me get this other one in here. Brewers use it in the mash all the time, all right? And for most part, a lot of the grains have these enzymes in here. So the base of our grain will include these starches or these grains that have amylases that are functional. And we'll talk about the temperatures of the mash Wednesday, I believe. All right? So we germinate, and then we have to stop it. How do we stop the grains? We stop them from utilizing all the starches by heating them and drying them out. All right? So let's talk about the grain types. There's no name on this. So we have our options on what type of grain we use. Not just like barley or grain or oats. But also in how hot we roast or how hot we dry the grain, and the temperature in which we dry the grain, and whether or not we do it on raw barley or we do it after we malt. So the general process for making these grains is to first malt the grain. So, and that's just the germination process and so forth. I think you had a video on it, right? Watch a video on malting? All right, so we've got our raw barley, and then we can malt it. Malting. All right? That's letting it germinate. And then we've got 2 things that we can do. We can dry the grain. And we dry the grain at about 140 degrees Fahrenheit. We dry the grain. The grain types that get this sort of treatment would include all of our base malt. Grab a different color. Could be all of our base malt. So it could be, let's say, Pilsner malt. What's called pale ale malt. So much better. Vienna. We've got Munich malt that does this. And we have some of the toasted specialty malts. Some of the toasted specialty. Now, when we dry it to get these malts, we're drying it at 140, but then we're going to kind of dry it the rest of the way in a kiln. And this is where we start to have a little bit of flexibility in our temperatures based on the characters. So what I've done, we're going to kiln it at 185 to 240. All right. So if we kiln it at this type of temperature, the lower temperatures will be these Pilsner and Pale Ale malts that are really light in color, little bit of flavor from the grain and from the toasting process. And then we go up slightly higher to the Vienna and Munich. We start adding a little bit more character because now the sugars inside of those grains are going to start to caramelize a little bit. They'll undergo various reactions to produce different flavors and aroma characteristics. All right, same thing if you've ever made caramel, homemade caramel. You caramelize sugar. How long it goes determines how much of that flavor really develops. And there's a range of flavors that you can get from it. All right, so we kind of cap out at some of these toasted specialty malts that are a little bit darker, but not anywhere as dark as some of these other ones. If, though, we go to a kiln at, let's say, 320 to 430, now what we're basically kind of getting to is more of a roaster. More of a roaster. This is kind of what coffee grains are doing. We still, have enzymes in it, or we still have sugars, but now we're really caramelizing those sugars. So now we can produce what's called chocolate malt. We get a black malt out of it, a special roast, and brown malt. So a lot of these, brown malt used to be very hard to find. It went out of favor. But brown malt was pretty common because early maltsters had a hard time trying to figure out the recipes, I should say the timing and the temperatures. It wasn't all automated, so a lot of times they over roasted them, got them in a little bit darker color. So more roastier flavors, darker colored beers come from this, and so forth. The chocolate and the black are probably the darkest of the malts. But we've been malted, we've been dried, all right, and then we kiln them to develop more of these flavors. All right, questions? We also have, so we did our malting, and then we let it germinate, and then we stopped it, we dried it. Instead of drying it, what we can do is to stew the grain. And then we can dry it and kill it. Right? So with the drying, we basically stop all the enzymatic reactions and let it work. Dry it, they save it. So in these early ones, Pilsners, Pale Ales, we can use 100% of that grain, and we're going to get a good mash. We're going to get all that conversion because the enzymes are there, they function. All right. In this stewing, what we are doing is allowing the grains to actually mash themselves as a whole grain. So the enzymes are going to be accessible. They are breaking down the sugars inside of the grain itself, and then we're going to dry them and we're going to kiln them. All right. This produces what we call crystal malts. And the crystal malts have different colors and have different flavor profiles based on the temperature and the length of our kilning at that time. The difference between these is that these are loaded with starches and our enzymes. This, it's called a crystal because what we've essentially done is converted the starches to sugar, and when we dry it, the sugar is basically crystallized inside of it. So these guys don't have any more starches. It's all been converted. These are just going to be pure sugar that add or contribute to character and the color. We couldn't really use, we could use 100% of these crystal malts, right? But we don't have to do a mash. The sugars have already been converted. Also, through the drying and the kilning process, we will destroy the enzymes. That's part of it's stewing, looking at 140, 150, right? But that's okay, because we already had the sugars. And the last bit is going back to this raw barley. Take the raw barley and roast it. So you skip the germination process. You skip the roasting, or you skip the germination process, you just start roasting it. This produces roasted barley. This produces roasted barley. Now the roasting process again destroys the enzymes. So if you use this roasted barley, you need to use it with some of these other malts to provide the enzymes. This guy has starches, has flavors, but doesn't have any of those enzymes. All right, so what we're going to be doing, we're going to do this after Labor Day on that Wednesday, is that I have a collection of these different grain types. And I'm going to do a miniature mash. It's called nano mashing. Right? It won't be alcohol, it'll just be sugar water. But what we're going to do is we're going to see the contribution of the different grain. You're going to not only see it, but probably smell it. And since it's sugar water, you can try it, you can taste it. All right, I'll have cups here that you can pour a little bit just to see the difference. I want you to see how we can mix in, we can use different types of grain to kind of really zero in on a specific color that we want. All right. And notice that these colors, we didn't even talk about the sugars, about the end product of the beer. We're just right now focused on some of the flavor contributions and our color contributions on these. All right. Any questions? All right, so that wraps up